PREDIVEZ vesicular transport kits provide all the necessary reagents and membrane preparations to enable the study of individual drug transporters using routine lab equipment. Before starting the experiment, the reagents are removed from the freezer and placed on ice to thaw. A serial dilution of the inhibitor is then prepared using the first row of a 96-well plate. This dilution can be carried out using the known inhibitor included with each PREDIVEZ kit or with another test compound in order to determine its inhibitory potency. The solvent is added first, and this is followed by the inhibitor, pipetting well to mix. The assay buffers are prepared by diluting the concentrated stock solutions provided in the kit with distilled water. Each buffer should be mixed well and then placed on ice until use. If included in the kit, the detector solution can be stored at room temperature. Once thawed, the ATP and AMP nucleotide stock solutions should be vortexed to mix and diluted using the appropriate assay buffer. These solutions should be placed on ice until use. The membrane suspension is prepared firstly by vortexing and pipetting the membrane stock to mix, and then diluting it in the appropriate assay buffer. The probe substrate included in the PREDIVEZ kit is then added directly to the membrane suspension. The suspension is mixed by inverting the tube several times and then placed on ice. The PREDIVEZ assay is carried out on a 96-well plate, which should be placed on ice while each reagent is added. First, the membrane suspension is added to the appropriate wells. 50 microliters of membrane suspension is added to each well, which corresponds to 40 micrograms of total membrane protein. Next, the serial dilution of the inhibitor or test compound is added to each well containing the membrane suspension, and the plate is pre-incubated at 37 degrees Celsius for 10 minutes, along with the diluted nucleotide solutions. The reaction is initiated by adding 25 microliters of the pre-warmed ATP or AMP to the appropriate wells. The plate is then incubated at 37 degrees Celsius for the time indicated in the assay protocol. During this incubation step, the filter plate should be prepared by pre-wetting all wells that are to be used with distilled water and sealing any unused wells to ensure an adequate vacuum. It's important that any water added to the filter at this point is removed prior to the end of the incubation. Once the incubation is complete, the reaction is stopped immediately by adding ice-cold washing mix to each well. The contents of the 96-well plate are then transferred to the filter plate. Vacuum is applied to remove any liquid, and each well is washed 5 times using the diluted washing mix. Following this wash step, the filters should be dried using a heat lamp or other suitable device. If using a PREDIVEZ kit with fluorescent probe substrate, the diluted detector solution should be added to each well of the filter plate and incubated for 10 minutes at room temperature. The liquid in the filter plates should then be transferred under vacuum to an appropriate 96-well receiver plate. The fluorescence in each well can then be measured using a fluorimeter. If using a radiolabeled probe substrate, scintillation fluid can be used instead of detector solution, and the amount of substrate present can be determined using scintillation counter.